In this presentation, we will be discussing both single-stranded and double-stranded DNA repair pathways.
There are three main kinds of damage for single-stranded DNA pathways. These include spontaneous mutations, pyrimidine dimers, and damaged bases.
Each of these three types of single-stranded DNA damage has their own DNA repair pathway. For spontaneous mutations, you have the mismatch repair pathway. For pyrimidine dimers, you have the nucleotide excision repair pathway. Lastly, for the damaged bases, you have the base excision repair pathway.
For spontaneous mutations, we use the mismatch repair pathway. This is a repair pathway for single-stranded DNA damage. The first step is the recognition of the damage by enzymes. Then they cut out the mutation and DNA polymerase replaces the nucleotides with the new sequence. DNA ligase then seals it together.
The pyrimidine dimers use the nucleotide excision repair. This is a repair mechanism for single-stranded DNA damage. This is done by three steps. The first step is the recognition of damage by enzymes. The second step is the excision of the damaged strand. This can be both upstream and downstream of the damage by endonucleases. These strands are approximately 12 to 14 nucleotides long. The last step is the resynthesis of the removed region by the DNA template strand using DNA polymerase and DNA ligase.
Base excision repair is a pathway used to repair single-stranded DNA damage. This damage is caused by oxidation, alkylation, hydrolysis, or deamination. There are two steps in this process. For the first step, DNA glycosylase removes the single damaged base. In the second step, DNA polymerase and ligase are used to resynthesize the base.
The three types of double-stranded DNA damage repair pathways are non-homologous end joining, microhomology-mediated end joining, and homologous recombination.
The first double-stranded DNA damage repair pathways we will discuss is non-homologous end joining. The damage is recognized by proteins, which results in the broken ends being directly ligated without the need of a homologous template.
The next double-stranded DNA damage repair pathway is the microhomology-mediated end joining. This works by ligating the mismatched hanging strands of a DNA molecule, removing the overhanging nucleotides, and then filling in any missing base pairs. This results in deletions flanking the original break.
The last double-stranded DNA damage repair pathway is the homologous recombination. This is where the double-stranded breaks are repaired directly after replication. This requires the presence of an identical or nearly identical sequence to use as a template, for instance, the sister chromatids.